Evolutionary biologist, and what it got me into biology were organisms such as these. So the things that have always fascinated me about biology is the breadth of diversity that we see. On the left here, we're thinking of in terms of taxa, different numbers of species. In fact, the one on the very bottom left is really what got me excited. So when I was a little kid, my parents took me to the Smithsonian and I saw fossils of dinosaurs for the first time, and that just really fascinated me. But my work is actually worried about things on the right, which is to say not the diversity amongst taxa and species, so much as the diversity of morphology and phenotypes. So we may see differing morphological or phenotypic traits amongst closely related species of cichlid fishes or various honey creepers, or different aspects of morphological variation in populations or species of flowers. And as an evolutionary morphologist, my goal is to try to explain this phenotypic diversity. Now, of course, to be able to do that, we need to quantify what we think of as morphology. And that in and of itself is a whole really interesting area that leads to what we know of as morphometrics. And I won't focus on that in the talk so much, but I will give you a little yellow brick road reminder of progress and how we have gone about quantifying things. And so, of course, simplest way is to break things up into categories. Humans are very good at doing this. And so large versus small, circular versus elliptical. And we can put things into different bins and categories of morphology. And that works pretty well until we get a third object that doesn't fit, right? And so then we move on and make multiple categories, which suddenly leads to rank orders. Small, medium, large. That's a good way to quantify morphology. And that works for a while. But then you want to know, is A and B, are those as different from each other as is B and C? And so that moves us on to counting things and being much more numerical about it. So maybe we count the number of scales or the number of seeds in a particular plant. Or maybe we pull out our calipers and we measure things. So here's an example on fishes where we measure lots of different traits with calipers, lengths and widths and heights. Those are very, very good at capturing morphology and phenotypes. But the problem with those are that you lose the relative location. So you could have the same length and width measurements from two different objects, but those objects have different shapes because the position of the length and width measurements are not recorded in those numbers. And so finally, what we move along to is something called geometric morphometrics, where we take these anatomical objects and we characterize them by anatomical points or curves or surfaces. So good homologous locations across objects. And this has led to a very rigorous characterization of morphology and the phenotype. Now, wherever you fall along this continuum of measuring your phenotypic traits, you can clearly see that what we go as we go from the left to the right, we gain biological realism. We capture phenotype more accurately, but this comes at the cost of being with greater data complexity. It requires some new mathematical theory, particularly multivariate theory, and it presents analytical and statistical challenges. And I'm not going to go into many of those today. I'll give you a few. But the point here is we've reached an era of big data, not just in terms of the size of our genomic data sets, but also our phenotypic data sets. So as a quick example, here's some data from a recent publication looking at various vertebrate skulls. And they're characterizing each skull by thousands and thousands of variables, though they might only have 100 or 200 objects. And so that high dimensional data problem is very common both in the genomic side of ecology and evolutionary biology, but also on the morphological, the phenotypic side. But what these technologies have allowed us to do is basically go from an object like this. So here we have a Caribbean gecko on a 3D scan of that gecko. And we can characterize that on the right with high dimensional landmarking and phenotyping. So if we think about high dimensional phenotyping, landmarks might be one way of doing that. There are other colleagues here at Iowa State that use other high throughput phenotyping. It really doesn't matter for our purposes today which of those types of procedures you're using. The point is we have really dense, high sampling of phenotypes as our quantitative measures of morphology. And our question is, how do we deal with that statistically? How do we retain statistical rigor to address the biological hypotheses that we care about? Now, for my purposes, I wish to take these sorts of data and apply them to macroevolutionary questions and hypotheses. So hypotheses about morphological change across species and across the tree of life, right? And so many times I will study salamanders or other vertebrates in my empirical research in my lab. And we're interested in various questions that evaluators seek to understand how phenotypic diversity evolves across the tree of life. And this requires two things: methods for quantifying these morphologies, these morphological phenotypes, and also statistical models for testing hypotheses in a phylogenetic framework. And those two foci have really been the bread and butter of my lab for quite some time. Those are the areas in which we have contributed a lot of our theoretical work. And so what I want to do today is briefly go through three components. I want to talk about morphometrics and phylogenetic comparative methods. And for the purposes of many of you in the audience, think about that first word, morphometrics, as just high dimensional phenotypes. Next, we're going to think about the macroevolution of those and give some empirical examples. And then I'd like to end with some current musings and thoughts about how we can compare data sets using effect sizes, multivariate effect sizes. Okay, so let me set up the problem from an evolutionary perspective. These are very common plots. If you go back into the classic literature from the 60s forward, you will see plots that look like this throughout the evolution literature. What you're looking at here are data where the data points are species, and we have various measured traits that are evaluated and compared and correlated. And these sorts of trait correlations across species were used to understand adaptation and to study coevolution. So finding very strong associations between body mass and metabolic rate was treated as evidence of adaptation and coevolution of those traits, for which explanations were required. But the problem that was recognized is that the data points, species, these observations are not independent of one another. And so what is actually required are tools that account for phylogeny, that account for that lack of independence throughout the analysis. And we call these phylogenetic comparative methods. So what these methods do under the hood is that they condition the data on the phylogeny during the analysis. And of course, our goal with this is to evaluate evolutionary hypotheses while simultaneously accounting for the fact that our observations are correlated. And doing this statistically turns out to be relatively simple if you know a few things. And the few things that you really need to know, you need to know an evolutionary model of how you expect variation to be generated, and you need to know the relationships of the observations, in this case, a phylogeny, okay? Now, I'll point out now, and hopefully I'll remember to do this again, I'm speaking at the level of interspecific patterns, correlated patterns. They need not be, their observations need not be species. The same idea would work at the population level for pedigrees, individual levels, et cetera. And so these tools are generally applicable to other levels of biological organization, provided a few analytical caveats. So what about the null model? Well, the null model that we typically use is Brownian motion. The properties of this are illustrated visually here. What you're looking at are a whole series of Brownian motion runs or evolutionary runs under Brownian motion. And what you see going from left to right is that the mean does not change, but the variance does. And in fact, the variance increases proportional with time. And if we use that as a null model, it turns out that statistically that falls into a very broad category of basic theory that is quite useful. So this is the continuous trait equivalent of your Markov process. This has connections to Gaussian theory, to the normal distribution. Anyone familiar with Galton's quincunx? It actually connects with that as well. So there's some deep mathematical roots to Brownian motion as a useful null model. And we use this as the model for evaluating trends in data while accounting for phylogeny. Now, how do we do that? It really depends on your question. So what I'm trying to illustrate in this slide is the fact that there is not one phylogenetic comparative method. I don't like it when people say I used the phylogenetic comparative method. In fact, that's not the case. The phylogenetic comparative method is a toolkit, and it has different tools depending upon the hypothesis in question that you're interested in. So we've got a set of related tools to evaluate phylogenetic signal in our data. If we wish to look at the association of traits via regressions and Anova, we have another set of related tools. We have tools that allow us to evaluate variance accumulation or rates of evolution and compare those. We've got tools for looking at that Brownian motion model and comparing it to an alternative. And so we've got a whole toolbox that is derived based on this notion of a null model for evolutionary change and a pedigree, a phylogeny for how the species are related. And it turns out that most of these are derived from the statistical model, PGLS, phylogenetic generalized least squares. The way that works, we've got our response data, our multivariate phenotypes, call that the matrix Y. And we're going to explain that. We're going to explain that by some hypothesis, biological hypothesis, which is X, our design matrix. Perhaps we wish to compare island versus mainland species. And normally you would do that in an Anova framework, but we know that the species, the observations are not independent, which gets worked into the error. Specifically, the error term incorporates what's known as the phylogenetic covariance matrix. It takes this phylogeny and converts it into matrix form that explains the relationships of the species. So specifically, the off-diagonal elements tell you how long species co-evolved together before speciation. Under Brownian motion, where change is proportional with time, that translates into understanding the expected correlations among observations. So it's a very elegant framework to incorporate the non-independence among our taxa. Great, so this is easy, we've got it. That math has been around for decades. Generalized least squares exists in the statistical literature and has been for a very long time. So why don't we do this? No problem, right? Well, to do this for high dimensional phenotypes, we need a couple of things. We need to condition the data on the phylogeny and get our parameters. We need a set of summary statistics of that fit. And then we need to evaluate that fit for significance and for the strength of signal in some way. And it turns out that for multivariate data and high dimensional data in particular, these are rather significant challenges to overcome. Now that sounds a little odd. In fact, and the skeptic would say, well, what are you talking about? We have matrix algebra that lets us handle multivariate data. We've got parametric theory. It provides us equations under likelihood. Just plug and chug. Okay, fine, let's do that. Let's do that for a very simple example of phylogenetic regression. I'm going to set this at 30 observations, so 30 species. And we're simply going to go from two-dimensional data and increase the number of variables. So our phenotypic matrix gets more and more complex. And we will simulate data with no relationship, and then simulate data where the relationship of X and Y gets stronger and stronger. So we have independent X versus Y versus highly correlated X versus Y. This will generate a power curve. And in fact, we have a whole family of power curves depending upon how complex the data set is. And what you see here in these gray circles is a nice power curve when we have lots of observations and only a few variables. But as the variables increase, as our multivariate data set, our phenotypes get more complex, power drops And eventually, boop, hit 0. We've got flat line. We are dead, statistically dead here, right? And so what this tells us is as our phenotypes get more complex, parametric methods begin to blow up. Why? Why does that occur? Well, it occurs for a very simple reason, log likelihood, right? All of our statistical theory is based on, parametric theory is based on likelihood equations. I won't go into the details of that. I can summarize the problem rather simply. In the denominator, we have this little bit, which is the determinant of V. V is that covariance matrix I had mentioned before, the covariance amongst objects relative to the covariance amongst the traits. And as the number of traits, which is P, gets larger, the determinant of V gets smaller. And eventually it hits 0. Well, that means we're dividing by 0, and math doesn't like to divide by 0. So put another way, as your data become more highly complex and more highly dimensional, the problem of parametric likelihood-based hypothesis testing becomes ill-conditioned, and we can't complete it. So we need another path forward. I would argue that what we wish to do here is, in fact, forego standard maximum likelihood and parametric approaches and instead use a set of robust methods. And the procedures that we've been developing in our lab embody 3 steps. First, we condition the data on the phylogeny and fit the model parameters. That step, model fitting, largely works as before, although I'll tell you how we implement very shortly. Second, get summary measures of the fit in such a way that we avoid this determinant equal to 0. So we don't divide by 0. And then third, evaluate significance and obtain the strength of the effect, not using the likelihood. Again, because the likelihood divides by 0. So how do we accomplish those? Now, the first step is actually relatively straightforward. There are conditioning on the phylogeny can be done in any number of ways. I will just visually give you 3. We don't need to concentrate on the details of the differences of the equations. One of them is phylogenetic independent contrasts. So the idea here is to take your species data at the tips of your phylogeny and obtain contrast scores at the nodes, which are effectively time standardized differences, which you can see down here on the bottom left. And then one uses these values in your analytics. So rather than using data for 4 species, we would use data for 3 contrasts. And that is one way to obtain model parameters. An equivalent way is through phylogenetic regression, the formal PGLS regression. This effectively takes non-conditioned data. Here's the error data before the phylogeny. And then after one has accounted for phylogeny, the remaining residual error is independent of it. As you can tell, it's just kind of bouncing around in this data space. That would work. Or a third way that's kind of elegant, and I like this one, is what's known as phylogenetic transformation. So what we do here is we identify a data space whose axes describe directions that are independent of the phylogeny, shown here in this plane. We take our data point, which is this black X, and project it into that data space. That gives us the red arrow. Now what we have done is we have rendered the data independent of phylogeny. And so this is converting a generalized least squares problem into an ordinary least squares problem. And at that point, we can fit the regression parameters just as before. Now, this one's been used for about 2 decades in phylogenetic comparative methods. We've kind of really made use of this in recent years. And that's what we do. All of those give us the parameters of a model. So that's great. That we can solve. We can do that for multivariate data. We can do that for high dimensional data. No problem. Step 1 accomplished. Okay, but what about step 2? How do we obtain summary statistics, right? Normally for multivariate Anova, you would do a Wilks lambda or a p-lies trace. Those are all going to blow up and divide by 0 as our data become very highly multivariate. So we can't do that. So we're going to use another trick from multivariate statistics. This goes back to Gower in the 60s. Actually goes a little further than that, late 50s as well. But Gower's paper is really quite instrumental here. What Gower recognized is visually shown here on the left. What you have in this first panel are the distances of each object to the centroid. And if you square those and add them up, you get your sums of squares. Right? And we're used to looking at sums of squares in Anova tables, for example. The left panel that's on the right panel of the left figure shows the distances amongst the objects. It turns out that the distances amongst the objects can be used to obtain the sums of squares based on the group centroids. In other words, there's a relationship between the distances amongst objects and the covariance that's obtained in the data set. And so we can calculate sums of squares using distances. If we do that, we can get the sums of squares of the model, the sums of squares error, calculate mean squares, get an F statistic. We can get summary statistics without dividing by 0. And so we avoid that determinant of 0 by using this alternative pathway. So this is akin to a Q mode versus R mode approach to statistical analytics. Now, we're not the first to have done this. If there are any ecologists in the room, Marty Anderson's Manova approach, permutational Manova uses this. In the genetics literature, the Anova analysis of molecular variance approach uses this. In Morphometrics, Goodall's F-test uses this. The point is, this is a nice way to get robust summary statistics without dividing by 0. So that's our second step. Now, significance testing, evaluation. Again, we can't compare to a parametric distribution because that's divide by 0 again. The determinant is 0. So we're going to use instead what is known as RRPP, residual randomization in permutation procedures. The idea is pretty simple. You fit your models. We fit the full model, which in this case, Y is being explained by X1 and X2. And we fit a reduced model. Y is being explained only by X1. We get parameters, summary statistics, etc., from the full model. We get the fitted values and the residuals from the reduced model. We then permute those residuals and add them back on to create what are known as pseudo values. Then we take those permuted components and run them through the full model to get summary values. And you repeat, and you repeat, and you repeat. And what you generate here, of course, is an empirical sampling distribution, not one generated from theory, but one that is empirically generated against which you can compare your observed summary statistics. Now, this is very useful again, because you'll notice we are not at determinant equals 0. So we're not dividing by 0 at any point. And it's been shown in the permutation theory literature that this approach to permutation using residuals is the most robust. And we've got a paper down here on the bottom that has talked about that. Kamanji's theoretical paper in 2003 was really instrumental. It talks about exchangeable units and what's the correct way to do these sorts of things. But there's one more really important piece with this. We have the observed value, and we have a distribution, a sampling distribution of possible values. The mean and standard error of that distribution allow us to obtain an effect size. So we can estimate an empirical sampling effect size for the strength of signal. And that's really useful because to me, statistics really shouldn't be treated as a light switch, on, off, significant, not significant. We also ought to interpret the strength of the signal. And this Z-score that we've developed, we feel allows us to do that. And I'll come back to that in just a little bit. Okay, so now we have our 3 steps that effectively avoid the divide by 0 problem with multivariate data and parametric likelihood theory. What happens when we put that simulated data through these procedures and look at that power curve? Well, power is just fine. We have broken what's called Raoult's paradox. So Raoult's paradox, I forgot to mention, Raoult's paradox, or the curse of dimensionality, was the observation that as you add more variables to your phenotypic data matrix, in this case, I have more information about my observations, but I have less statistical ability to say anything because the power goes down. We've broken that. And in fact, power in some cases can go up with this procedure, because anytime you add a variable that contains a piece of the signal under investigation, it'll pick it up in those distances, but not at the cost of extra parameters in that covariance matrix that requires inversion. And so these approaches seem to have done the trick in terms of statistical evaluation. They also estimate the parameters of the model properly. And in fact, you can ask whether or not this procedure matches parametric likelihood theory. And we do that by comparing theory with RRPP in those cases for which both can be calculated. So in other words, we use lots of observations and just a few variables. So where we know both are robust. And here's one example. What you're looking at on the right is the theoretical F distribution for a N of 128 and 5 variables. And that's in black. And in red is the theoretical sampling distribution, the empirical sampling distribution from permutation. They are perfectly overlapping. So what this tells us is, in fact, this RRPP approach, when applied correctly, is identical to likelihood and parametric theory in terms of outcome. It is not a substitute for under bad conditions. It is parametric theory, just obtained differently. And in fact, that's a conversation for another day. Statisticians in the 40s and 50s understood that permutation and, in fact, simulation-based inference were useful ways of generating sampling distributions that match the ones derived from calculus. In case you don't believe it, and we had a reviewer number 3 at one point that was still skeptical. They said, well, really, you ought to go into multivariate theory and pull random covariance matrices and look at their distribution. Okay, that would be by drawing samples of covariances from a Wishart, which is panel B. And here is the distribution. It is circular. Here are the empirical covariance matrices on the left through RRPP. And in fact, we have less wiggle and wobble, eccentricity, from RRPP than you get even from sampling from the theory. And so I might even argue we're better than theory here. The point is, if we use this permutation procedure, RRPP, and combine that with robust summary statistics, we have a path forward for doing multivariate phylogenetic comparative methods. And we can do so in a way that doesn't break the bank. We don't blow up and divide by 0. And in those cases where parametric theory and likelihood could be calculated, we converge on exactly the same answer. And so we contend this is a useful solution. Now, what we've done with that, since these insights, we've gone back to that comparative method toolkit, and we have generalized the parametric tools for multivariate data. And so we've generalized a series of tools for phylogenetic Anova and regression under this framework, for multivariate correlations under this framework, so-called phylogenetic partial least squares, for comparing evolutionary rates, for looking at multivariate phylogenetic signal. And so we've systematically managed to find generalizations of the parametric tools to handle and incorporate multivariate data. And so this facilitates investigations of macroevolution for complex phenotypes, whether those complex phenotypes are shape or suites of measured variables or suites of behavioral or physiological traits or functional morphological traits. This really opens up the door to be able to address similar questions that we've been doing for quite a few decades now for univariate single-valued traits. Now, there's one other little advance I'd like to mention here, and that deals with visualizing the data. So any of us who have multivariate data sets have a problem with visualization. Maybe I've measured 12 variables. I'm pretty good, but I can't see data in a 12-dimensional space. So we need a way to summarize that. And if we have data, interspecific data, traits measured on species, we also would like to incorporate the phylogeny. And the way that's typically done is using a variety of principal components analysis. So principal components analysis will spin the data around to directions of maximal variation based on something. And the standard way to do this is naive principal components analysis, and then you project the phylogeny into the space. And that's called a phylomorphospace. You have an example of this on the left here. And so that's just a basic PCA with the phylogeny projected into it. A second procedure is called Phylogenetic Pca. And this aligns the data. It's slightly different algebra. But what it does is it aligns the data to directions that are independent of the phylogeny. And in particular, the first phylogenetic PC axis is independent of phylogeny. And I need not go into the details of why that is the case. The point is it gives us a different picture. So if I look at my data here, I've got two different plots to view this data space, both of which are mathematically reasonable, but my interpretation gets challenging. And one reason it gets challenging is because I don't know how much of the data is, say, ecological signal versus phylogenetic signal, right? You don't know a priori the extent to which different components of signal are mixed in our data set, in our phenotypic data set. So what we just recently proposed is a third ordination that we call Paca, phylogenetically aligned component analysis. And so what we do instead of partialing out the phylogeny, we're going to rotate the data to the phylogeny. So we're going to align the data in directions that maximize the phylogenetic signal. And the simplest way to understand what it's doing is, again, visually. So on the left, you see a phylogeny. I'm denoting the species clades in blue and gray. And then there's circles and squares for two different ecological groups. And if we simulate data that's only phylogenetic data, what we get are phylogenetic Pca and Paca plots that are similar in the respect that the blue and the black dots are separated. Okay, they're not aligned identically because the algebra is slightly different, but both of them manage to separate the groups. In the case of Paca, it's along the first axis. In the case of phylogenetic Pca, it's actually closer to the second axis because that first axis is rendered independent of phylogeny. But what happens when we create data that has both phylogenetic and ecological signal in it? Well, now Paca still captures the phylogenetic component of that signal. But phylogenetic Pca, notice what you have here. On the left side of PPC1 are all of the circles, and on the right side are all of the squares. So it actually has separated out the ecological signal. And so what we argue is that using Paca in conjunction with something like phylogenetic Pca allows one to get much closer at revealing the relative components of signal in your data, whether there is a phylogenetic component or an ecological or both. We could follow that up with some statistical tests as well, but this provides a very nice visual for what's going on. When both plots are similar in terms of separating out major clades, that tells you that the phylogenetic signal is quite predominant in your data set. Whereas like in this bottom plot, when Paca and phylogenetic Pca are very different, some other aspect of signal is present in your data set. Okay, so these are the kind of tools that we have been developing in the lab the past decade or so. And I want to take two examples, I think it is, just to show you how they work. And I realize this might be a little bit far afield from the current audience, but bear with me, vertebrates are beasts too. And so I work on salamanders, or at least I have for much of my empirical life. And plethodontid salamanders in particular are quite an exciting group to me. They have a worldwide distribution. Most of them are North or Western Hemisphere, but there's one species in Korea, and there are 8 in the Mediterranean, primarily in Italy. That's another story for another day. As a morphologist, all you need to do is look at these pictures around that I've provided here. There's a diversity of morphologies and phenotypes in this group. Many times those are associated with different habitat types. So some of these species live in terrestrial habitats, other in aquatic habitats, and a few other things. And so it's a very rich group for us to evaluate why phenotypic diversity has evolved, why we can evaluate hypotheses for why this might have been generated. And so what we've been doing in the lab when we get to that stage is we use these multivariate phylogenetic comparative methods to test hypotheses of morphological change across the tree of life of salamanders. And so one quick example I'll give you. Oh, by the way, so this is me in a cave in Tuscany. So I did a sabbatical many years ago to work on those salamanders in Italy. Nice thing about field work in Italy is when you emerge from the cave, you can take all of your cave gear off and go to a wonderful Tuscan restaurant. So that part was not so bad. What's cool about the salamander species there is that they are primarily cave dwelling and they cling to the ceilings and the walls of the caves. And they do this with a rather interesting foot morphology. And so one observation that we made was that if you look at the ontogeny, the growth trajectory of foot shape, which you see here on the right, what you notice across species is that the juveniles are quite different. That's shown in blue. The juveniles across species, among species, are quite different in foot morphology, but the adults are much more similar. And in fact, a permutation test shows that they're significantly more similar as adults than they are as juveniles. So this is evidence that there's convergence of adult foot morphology, which is consistent with a hypothesis that there's strong selection on foot morphology. Second piece of evidence bolstering that hypothesis is shown here. Here we're looking at 3 populations, one that's primarily terrestrial in the forest floor, one that occupies a natural cave, and the third has recently colonized an artificial cave. And we know this because this was a cave built during World War II to serve as a bomb shelter. And what we see on the right is that the foot morphology, in this case foot area, is again converging on a common cave dwelling morphology. So again, invasion into this habitat elicits a phenotypic response. So if we put all of those together, that plus behavioral data suggests that there's rather strong stabilizing selection on foot morphology. They need to have a certain morphology or frankly, they fall off the walls. And so that generates a hypothesis. I think my computer's frozen. Let's try this again. Are you still there? My apologies. We have another computer difficulty here.
But it's okay. Yeah, we're here. It's stay on this content.
It is stuck on that slide. Yep, my computer is spinning and spinning and spinning. Gonna have to close it. I do apologize.
Okay.
Incidentally, the next computer is waiting. It has been ordered. Let's try this one last time. All right, so here's the question. If there's stabilizing selection, if we zoom out across the tree of life, we might expect lower rates of phenotypic evolution in traits related to foot morphology. And so what we did was we took these multivariate comparative methods and we looked at rates of phenotypic evolution of these traits. And on the bottom left, you see, oops, you see the body traits.
I think we're not seeing the slides yet.
You do not see the slides yet?
Yes.
Oh my goodness.
Oh.
I apologize.
That's okay. I thought you were gonna just feel the, time.
Why do we not see those?
Okay.
Oh, I see. There we go. And give it a second. Come on, come on, come on.
Yeah, it's coming. Yeah.
Okay.
Wonderful. It's coming. Yeah.
Man. Technology. Okay, we see a slide now.
Yeah.
Okay.
Wonderful. Good.
So, bottom left, rates of evolution for traits not associated with climbing, relatively high. Middle bottom panel, foot traits are lower. And so, yes, we actually see lower rates of evolution in these foot traits in those species that are climbing. Now, reviewer number 3 said, well, wait a minute, maybe all foot morphology is evolved slowly in salamanders. So we looked at another genus, performed the same test. This was a genus that is terrestrial, doesn't inhabit caves, and their foot morphology is on the far right here. And again, we see it's on par in terms of its rate of evolution with the body traits. So we've got the negative and positive control for this evolutionary hypothesis. And so yes, indeed, we can infer using these tools that the rate of evolution of foot morphology is reduced in this lineage of salamanders. Now, I mentioned these organisms live in different habitats. And so very briefly, one interesting habitat that they live in are caves. And you can see this in the green on the phylogeny. They've actually, or not caves, trees. They have invaded the arboreal habit at least 6 times over the history of this family, which is a kind of bizarre place for a salamander to live, particularly if they are plethodontids, which are lungless. They breathe through their skin, and they might be exposed to greater desiccation. So we might want to ask whether or not this shift to being an arboreal salamander has resulted in a unique morphological change. In other words, is there an arboreal phenotype? That's actually a rather common question that we ask regardless of what organism we're looking at, that they've invaded some new habitat, they might have adapted to this new environment. Has it resulted in a consistent phenotypic difference? And so we can apply the same set of tools that I developed, the multivariate Anovas and PCAs in a phylogenetic framework. And the short answer is no. There's no evidence of a unique arboreal phenotype. You can tell that simply because of the overlap in these PCA spaces. The arboreal in green is covered almost entirely by these other groups. So they've not shifted into a new region of morphospace. And of course, our phylogenetic Anovas also provide evidence that that's the case. We do, however, see lower rates of evolution in that group. And so perhaps evolving to the arboreal habit hasn't created a new phenotype, but it has slowed down the evolution of phenotypes in general. And so those are the kinds of things that we can evaluate. I think in the interest of time, because we had some technical difficulties, I will just skip this last one. We had some other examples here that I wish to show you. The last piece I want to mention for the last few minutes here is to talk about the strength of signal. And this is current work that we're doing. I would make the argument that all of ecology and evolutionary biology is really about comparing things, right? Obviously, phylogenetic comparative methods, it's in the name, so that's inherently obvious. But we do this in ecology as well. We quantify the strength of desiccation resistance in some population, and then we might wish to know whether that is similar in another population, or whether it's similar in a different environment. To do that, we need to be able to have summary measures of the signal that we're talking about. And so I'm illustrating here is a common measure of signal. This is a type of correlation coefficient, a multivariate correlation. And these are generated from random data. And this shows a problem with comparing summary measures. So what you see here is that the expected value of the summary measure decreases with sample size for this particular summary measure and increases with the number of variables. Well, that's bad because if I go out into the field and measure two populations, I'm not guaranteed to have the same sample size, right? Or I could flip this around and be a little cynical. If I want a high correlation, I either measure lots of variables or very few specimens, right? So this isn't very good. We shouldn't really be comparing things with this sort of a tool. And so we need something else. And I would argue, going back to the RRPP, the permutation method, that in fact, we can do this. So here's that same set of data and that same correlation coefficient. It's a PLS, partial least squares correlation. And what I've done in the bottom panel is simply run the permutation tests and obtained the effect size, the z-scores for each of those thousands and thousands of simulated data. And what you see here is while the actual summary test measure varies with n and with p, the z-scores are robust. They do not. And in fact, the z-scores in these simulations were at 0, which makes sense because I simulated data with random correlations. And so by converting our data to effect sizes, this actually gives us the ability to compare across data sets or across systems. And in fact, we contend this is a pretty general solution. And in fact, it goes all the way back to meta-analysis. So a little digression for you. Meta-analysis, the classic paper actually came out from Glass in 1976 in Science. He was interested in combining data across published data sets. And he advocated that you do this by combining effect sizes, right? And the quintessential effect size is the z-score, right? It's the first equation we learn in biometry, right? You've got the mean minus the expected mu and the standard error. Well, we can do this, but with RRPP, we use not the theoretically derived mu and sigma, but the empirically derived values from the permutation procedure. So we estimate, we run our data, get our summary measure, and estimate Z through the residual permutation procedure. We do that for two different data sets. We then run and calculate the difference between them, just using a two-sample Z-score. And now we have a statistical test that allows us to compare the strength of signal. But this is the strength of signal in multivariate data. And to my knowledge, this is the only procedure that allows us to compare the strength of signal in multivariate data sets, even when the number of variables is greater than the number of observations, right? So when we're in the true high dimensional data range, we can do this. And this is pretty general. So for example, maybe we've got a factorial model. We want to know whether phenotypes, variation in phenotype is explained more by sex or by population or by some interaction term. Well, look at this. We've got z-scores down here. We could evaluate and compare those z-scores. That's a path forward to model comparison in the multivariate framework, right? We could compare different levels of phenotypic integration. So we can envision scenarios where we want to know whether the morphological correlations, the integration is stronger in one data set as another. So one common thing is looking at the effect of environmental perturbations. So oftentimes we think that pristine habitats, organisms develop rather straightforwardly, whereas in environmentally perturbed habitats, there's developmental instability that leads to a reduction of morphological integration. We can do that. And in fact, we have done this. This is some data from lizards. Interesting, in this case, the biological hypothesis was not supported at all. So we have rural lizards and urban lizards. And the urban lizards were expected to have lower levels of morphological integration, and they do not. All we did was reify the fact that juveniles have less integration than do their adult counterparts. So we basically reified developmental canalization. So sometimes the hypothesis we are investigating goes that way, and that's just kind of the way science is. But the point is we have a tool for comparing those signals. We have a tool for comparing the opposite, modularity. What if your phenotypes had subcompartments, modules that are highly correlated within the modules, but less correlated between them? Well, I've developed a covariance ratio, a statistic for measuring modularity, and using these z-scores, we can compare different hypotheses of modularity. So here we've got the skull of a mammal, and you can break it up into 1, 2, 3, 4, 5, or 6 different modules. And there's different hypotheses in the literature based upon developmental considerations or genetic considerations or functional considerations. We can test them all against one another and ask which of these hypotheses of modularity best describes the signal that we see. And again, we use the z-scores. We can even use these z-scores for comparing the levels of phylogenetic signal. So we very recently adapted this tool for comparing the strength of signal, a phylogenetic signal in one data set versus another. So what I basically am contending here at the end is that using an empirically derived effect size, a z-score from permutation procedures, from proper permutation procedures, provides an analytical path forward to a very long-standing problem in biostatistics, which is how do we compare the strength of signal across data sets in some reasonable fashion? And yes, I know we do have some tools that let us do that in some contexts, but it's hardly a universal tool, and it certainly fails when we hit multivariate and a high dimensional problem. Okay, so I think I will wrap up before my computer decides to stop again. There are 2 take-home messages of my talk today. The first is that if we wish to evaluate phenotypes, multivariate phenotypes in a phylogenetically informed way, the combination of residual permutations and phylogenetic transformation allows us to get there. We use robust test measures and permutation procedures to do so. And secondly, we can leverage that same permutation regimen to obtain an effect size that gives us the strength of that signal, which we then can compare across units, across experiments, across species, populations, et cetera, which really lets us get back at good comparative strong inference. And so I'm gonna stop there. I have lots of folks that I would thank: students, postdocs, collaborators, funding sources, the teams for our 2R packages, Geomorph and RRPP. But I will stop there and take questions should we have any more time for that. There.
Wonderful, thank you very much, Tim. Yeah, we have open for questions. People that, you want to leave early, ask questions early, and then people stay around for another 30, 40 minutes. That's perfectly fine.
So this is Bruce Walsh. I have a question and a comment.
Okay.
So it strikes me that the dimensionality problem is arising because your variance covariance matrix of your traits becomes singular because you basically measure things which are projections of other things. And so what about taking a singular value decomposition and then truncating it for the traits you measure at some small value, singular value, so you have a reduced matrix, and then using that? Because the idea, you're measuring so many things that some of the things you're measuring, at least in your data, are perfectly predicted by other combinations. And that might be one way to get around this dimensionality issue, because in some sense, it's a false issue. You're essentially have a multi-dimensional projection. You're measuring more things than there are dimensions in that projection.
Sure, that is a classic procedure that has been used for decades in multivariate analyses. Use your singular value decomposition, project out the offending dimensions at the end that generally are traditionally explained as, oh, there's a little bit of noise here, or the redundancies have gone away. And that's absolutely true. And one could do that. It is also known, however, that comparing objects in the reduced space to the full space does not result in perfect correlation. So you are missing information when you do that. And taken to the extreme, that gets folks into trouble. So, you know, in our field in evolutionary biology, it's replete with examples that follow this pipeline. Obtain phenotypic data, perform discriminant function analysis, get scores along the first projected discriminant axis, then perform Anova to see whether my groups differ. Well, that borders a little more on inanity, because what they've done is they've projected out so many other components of the data, you're guaranteed to get it in that case because that's a canonical ordination axis. Using the first PC, again, I'm making it extreme, the first PC only implies that the direction of maximal variability coincides with the direction of the signal that one is trying to investigate. And that need not be the case either. So it's absolutely... Mm-hmm.
The classic is for, you know, as morphometrician, the class is for height. But I guess to bring it to a discussion for the more broad audience here is, sure, if I'm doing high throughput typing, I'm measuring everything I can think of. And I'm measuring it as a time series. So I have a series of covariance matrices going through time, basically a tensor. What's the best approach to extract information from that so that a breeder can very quickly pick out the components they want to follow? Obviously, there are certain things you want to follow, but there are other things you're trying to use to predict other traits of interest they can't measure directly. So what would be your insight on that from a morphometric standpoint?
Wow, oh, that's a great question. I think I would need to throw a battery of tools at that to extract that. So one thing that comes to mind here is you have your design, your time series design, a redundancy analysis, which is akin to what you're describing, Bruce, of you know, projecting into a lower dimensional space, but it does it in a model-informed way. That might actually get closer to directions of phenotypic interest to the breeder, right? And so a constrained ordination type procedure might allow you to get down and reduce to some of the essence directions. I'm not sure whether any of the factor analytic models have been extended to time series. They may have been, I do not know, but that might be another path also.
Well, I see that Jack Deckers is on here. And so the animal breeders have for quite a while used, Kirkpatrick called them infinite dimensional characters, but it's also random regression models in animal breeding. And that, I think, something important to bring over plant breeding, because it basically handles a series of correlated characters changing over time. And plant development as such is a lot easier to predict than animal development in some respects.
Absolutely, yeah. And those infinite dimensional models, the so-called function valued traits, those actually tie back into the piece I didn't talk about today, which is geometric morphometrics. You can think of those as trajectories through time, high dimensional trajectories through time. And there was a paper a few years ago by Goolsby in systematic biology that actually used some of the procedures I developed elsewhere that kind of links in with those. Yep, great question, thank you.
I'll ask one while everybody's thinking. So when you actually explain this RRPP in terms of that classical paper people published earlier about exchangeable units, Gruer 1966, it just seems like so straightforward, but I don't know why in the many literatures there was earlier permutation test, you know, ask people to permute the Y side or the X side. The Y would just, you know, it seems like permute the residual, except you have to set a model first, and then permuting the residual, get away from disrupting any phylogenetic or pedigree relationships matrix just right away. So do you have any insights why that has to be revealed later on?
Yeah, I do actually. So this is something that we're currently trying to summarize because it has been a rather convoluted history in the permutation theory literature. Part of it has to do with the fact that different groups were trying to propose permutation schemes that isolated the variance in what's known as an exact framework, right? So if you had a factorial model, you're looking for the exact variance components, and so therefore you could permute it this way. Or, oh, nope, this is a nested model, so now you actually have to do it this other way. And so you end up with, there's four main ways, right? You can permute the raw data, the Y data, the raw data. You could permute the Y data restricted, like within little subunits.
Yeah.
You can permute the residuals in various ways, or you permute the X variables, the independent variables.
Yeah.
What's been shown since is that across models, so across different statistical designs, and both for independent and correlated observations, only residual permutation works in all of the cases. And so this is one that, so that's been shown for ordinary least squares, independent. We've just finished simulations showing that for generalized least squares. So we're trying to write a big synthesis to let that people know that, yeah, there's all these different ways of doing it. This one way actually does it for all of those designs, for at least for linear models.
Right, that's right. Thank you. That's very clear.
Yeah.
So Dean, this is Jack Deckers. Really appreciate the talk. Very, very interesting, and methodology that I hadn't thought about. But we are running in these same problems also in animal breeding and in plant breeding, right? But in that case, we have we're not looking at observations on different species, but on different individuals. But we could have high dimensional phenotypes, right? Just like you have on the species. So, and my understanding from what you were describing, so rather than using phylogenetic derived relationships, you could use pedigree or genomic relationships instead. But then the question is, well, how large of a data set could it can the methodology handle? So not just in terms of the number of phenotypes, but also the number of individuals that you have those phenotypes on.
That's a great question, Jack. Because there's in theory, any, and then you hit, then you run into the problem that your computer just crashes like the brick wall behind you. Yeah, those matrices get very large in a hurry. And in fact, so two pieces. So I'll come back to that. The first piece is you're absolutely correct. You can use pedigrees. You can use phylogenies. The same analytics will work for time series. So things are not independent by time. That's another kind of covariance matrix, or spatially. Any of these things generate a generalized least squares framework. These tools are appropriate. But you're going to end up with a memory issue. We just, this week, actually, put out new versions of both of our software packages that make use of sparse matrix calculations. And so basically, if you think about a pedigree, it's a huge matrix with a lot of zeros. You don't need to be doing calculations where you multiply something by a bunch of zeros and add it all up. You just have to multiply it by the bits that matter. And so we've taken a few steps forward in the R packages we've developed just now, actually, to try to make use of that. And that will allow larger data sets, both in terms of observations and variables, to be handled more efficiently. We've not yet pushed them to their limit, so I can't give you a final number on N or P, but we are thinking clearly in that direction, because we'd love to go total big data with these tools, should computer memory allow it.
Yeah, that's the sparse matrix methodology is used a lot in animal breeding, mixed models, and yeah, with pedigree it's very sparse. But then now that we have genomics and we create genomic relationships, they're not sparse anymore.
Sure.
Because you can't have your cake and eat it too.
No. That's right. That's right. Yeah. If you describe the correlation of observations with something that explains everything, then you can run the model and not get very far. Yeah.
Good.
Generally, if people want to turn on the radio, they can go ahead and ask questions. We had quite a few students actually very active participating. I'll ask another question. So when you actually show the contrasting plots about comparing the test statistics in terms of the increase in size of the N, increase size of the P versus the Z-score, right? That's also kind of striking. You actually cited a Glass 1976 meta-analysis paper to justify what you're doing. So do you know why people
I have a question, Dr. Adams.
Embrace the Z-score?
I'm a student of online plant breeding course in Iowa. So my question is like, so especially in the plant breeding, like if we have more data observations, like the precision is so high normally. Like let's say if we think about the heritability or something, like if we have more data, like normally we get good prediction or heritability. But you said like in the likelihood function, like if we have more data, like it's going to zero, like, and we face some kind of difficulty. So I'm just wondering, like in the plant breeding concept, how we can relate that one.
Okay, so perhaps, yeah, I'm sorry, perhaps there was a piece for which I was not very clear. You can think of your going big data in two ways. One is increasing the number of observations, which in my example would be the number of rows. And then one is the number of variables, which in this case would be the number of columns. If you're looking, let's take just a single variable, so one column, and to estimate your heritability, you need to get your variance components. You will have greater accuracy in those variance components by increasing the number of observations, right? And so from one perspective, yes, that's exactly correct. The challenge I was trying to explain is when you add more variables, that's when power goes down. So yes, you're correct. You get more accurate predictions of, say, a single trait heritability by increasing the number of observations. When we increase the number of variables, I have more information about the overall phenotype, but less ability to answer things with it because of this power dropping issue. Does that help explain? I'm sorry, I wasn't very clear about that during the talk. Does that help clear it up?
Yeah, thank you very much.
Oh, you're very welcome. Yeah.
Yeah, my question was just, can you comment on why, instead of using a, specific test and statistic, transforming that to Z-score, you have a constant flat line that you don't change. Yeah.
Right. So the basic idea there, again, goes back to meta-analysis, where one is attempting to consolidate sets of results into a single result. And just getting the average F statistic is not overly useful because the observed F statistic depends on many different components of the experiment. Even looking at correlation coefficients, the average correlation coefficient across studies, it's constrained. It goes from -1 to +1. They must be comparable. Well, not really, because the same strength of signal could be found, could generate a different correlation coefficient depending on sample size or some other component. So what the meta-analytic literature recognized was if you convert your test statistic into an effect size, that puts things on a comparable scale, a scale against which we can compare the values. And so what I'm attempting to do here, what I'm arguing is that we can do that for multivariate data as well if we make use of permutation theory.
So Dean, are those treated as fixed effects because there's, three different schools of meta-analysis?
Right.
Fixed effects, random effects, in the mixed model. And it's the mixed model may be more appropriate. You have certain things like sex effects, which are fixed, but then random effects would be, phylogeny or things like that.
Yeah, Bruce, you're absolutely correct. For the moment, since we're just in the nascent stages of this development, we're doing it just with the fixed effects, and then we'll be generalizing that on to random and mixed models. Just as incidentally, if I think back to the meta-analytic literature, that's kind of the way that developed as well. Yeah, but we're not quite there yet.
So to your final slides, you show the z-score of RRPP that, you know, instead of just grab whatever statistically reported, you have to go to the original data to do the permutation to get the z-RRPP and then summarize them together, right?
That's right. That's right. So the idea is we have some observed test measure from our data, multivariate F. We permute the data many times, and each time we get an estimate of that F statistic that provides us an empirical sampling distribution from which we can get the mean and the standard error, and then those are used in the z-score. That's correct.
Okay, thank you.
Yep.
Dean, I have another question. So is an alternative to address this problem of a P larger than N using Bayesian methods and MC-MC sampling? Is that an alternative strategy to address this?
For some applications, absolutely yes, Jack. Yeah, that is the case. One needs to be somewhat careful, however, if in that pipeline are likelihood calculations that require the full covariance matrix, right? But yeah, that is another route that some folks use to circumvent these sorts of issues. Yeah, yeah.
In one of the example, you showed that about Paca, introducing the Parka analysis, right?
Yes.
I'm assuming that when those data was not collected to show this phylogenetic relationship, they're just partial some data. Now you try to see if the Paca can separate the ecological and the phylogenetic relationship into, you know, rice shape, and not, you know, just partition that into appropriate forms, right?
That's right. So in the case of Paca and Phylogenetic Pca, we have the phenotypic data set, and we have an estimate of the relationships. We have an estimate of the phylogeny. Those are the inputs. And then given those inputs, one rotates the data either to the phylogenetic signal or away from it. So we're not in those procedures, we are not estimating the relationships of the phylogeny of the objects based on the data. We already have an estimate from other means. Yeah.
Yeah, those other means of the phylogenetic relationship, that's like a given. You just set that as, you know, not change that, right? You're not to challenge anything on that, right?
Correct. Yep. We take that as given and don't alter it during those analyses. That's absolutely correct. Yes.
Hi, Dean. Great talk.
Hi.
I'm Paul, a student with Dr. Stephen Cannon. Wanted to ask just a general question on the science of phylogenetic analysis as they relate to, I guess, plant species and yeah, animal samples. So for those cases where there might have been exchange, you know, between in genetic material between these individuals, do you have any thoughts on how using a bifurcating sort of display like a phylogenetic analysis would be a problem for viewing relationship among these groups?
Sure. Yeah. So genetic exchange by various mechanisms can challenge phylogenetic inference. And so what is commonly done, at least with genetic data, is one uses many markers, and then there are concatenation methods. There are different approaches. Either you use a rather complex Bayesian model where each gene is allowed to evolve with different parameters and rates, or there are some other concatenated methods where you estimate phylogenies from each of your genes and then concatenate them in some way. Those are paths forward, but there are certain cases where a bifurcating relationship is just the incorrect way to think about it. So for example, let's not go between species, but within. So if we look at populations, say 10 or 15 populations within a species, the genetic relationships are best represented by a matrix, right? Because you've got gene flow that could happen amongst more than 2 populations. And so then it's really, it's represented visually as a network, but mathematically to me, it's just the same. It's just a matrix, right? But a phylogeny, a phylogenetic covariance matrix will have lots of zeros where there's, no branches, as opposed to a network or a migration matrix, which would have presumably non-zero values due to migration. So yeah, you're absolutely right. Forcing bifurcating relationships in systems where they are not the case could lead to the wrong covariance matrix that gets used in these procedures.
I see.
Very nice question. Thank you, Paul.
Could I ask maybe just one more?
Of course.
P-values have been presented as somewhat controversial in deciding significance. Do you have an argument or an opinion for just using effect sizes to replace P-values in that case?
It's a shame you weren't in my statistics class yesterday, because I talked about model comparisons and we spoke a lot about this. I am a firm believer that one should not just use significance levels as if they were a light switch, right? Oh, it's significant. Oh, nope, it's not. One should always accompany significance tests with an effect size to aid in the interpretation. That's my perspective on them. I do not subscribe to the camp that you should abandon significance tests. I don't think I think that throws the baby out with the bathwater, but instead join the significance testing with an effect size to interpret it.
That's a good point. Thank you very much.
You're welcome.
Good to hear your short answer, but I like your answer. We always talking about pattern, we talk about signal and noise. I can't take that, the other argument saying completely abandon the p-value, right? How do we separate the noise with signals, right?
Yeah. There's some interesting history about those also, but yeah.
Yeah, that's wonderful. I think when you giving the lectures and then have you talked to the colleagues in genomics, do they complain about, typically we use the P parameters just for the X side, especially when we do this genome and phenome relationship.
Oh,
in your case, you use a P for the Y side, right? So
that's right.
To explain this whole thing, right? I mean, you can, like you said, you can do both sides, right? How do you think about explain that to people?
Yeah, I guess it's fortunate that most of the talks that I give are not to groups where P is on the X side, P is on the Y side. But sure. I would have to, you're right, I'd have to qualify that one. So, okay.
But then in your talk, you also mentioned that, the Y can be, this measurement or the other measurement, can be any measured variables. Have you actually tried to, let's say, there's some specific genes that measure the haplotypes. Now you on the Y side and still, have a phylogenetic analysis.
Right.
Some sort of analysis. Yeah.
I have not, but that is theoretically possible, and it requires a pre-processing step. So all of the methods that I have developed assume that the Y, the response variables, are multivariate normal.
Yes. Yeah. Okay.
So what you would do with a series of haplotypes is you would take your haplotype data, convert them to a distance matrix using the appropriate distance measure. And then if that distance measure is metric, you can use that in all of our analyses.
All right. Yes.
And in fact, the software that we've developed, RRPP, you can just give it the distance matrix. Or we can talk about this perhaps at length. What we do is we actually use principal coordinates to create a set of continuous variables that get used. So yes, you can.
Okay. Yeah, I know you have to leave at 4:30. Is that right?
Yes. Yeah, I do actually.
Okay.
So
if there are still students here, can you turn on your video, please? Let's give a round of applause to Dr. Dean Adams. Thank you very much. Wonderful talk, inspiring.
Thank you all for your attention.
Yeah, you can take off, Dean. I'll talk with some students if they want.
That sounds great. Thank you very much.
Oh, sorry, we didn't get an opportunity for you guys to ask questions. Matt, I mean, I hope that some of your high throughput phenotyping or measurement of roots or something can be multivariate on the Y side, or I think Clayton still here? Yeah. Is that something relevant to you guys?
It is.
Yeah. Go ahead, Matt.
To be honest, like I was sitting there trying to like Google a lot of the stuff he was talking about.
Yeah.
And so there's a bit of like, I want to ask questions, but there was a lot of just trying to piece together parts of everything that he's putting together with some of the, like the math.
Oh, it'll take some time. Yes. Yes. Yeah. Clayton, go ahead. Yeah.
Yeah. I think for me right now in my analysis, this whole conversation opens up a lot bigger world that I hadn't really investigated. And so like as of right now, we've been investigating like the root traits independently or looking at single genes. And this really opens up the possibility of saying, yeah, we could investigate the whole gene series or a whole complex of traits. And I think for me, where this really applies is as we start to look at the whole root shoot relationship and bringing in the phenotypic above ground and below ground relationships.
Yeah,
I mean, that multivariate value is then it becomes exponentially huge.
You talk about this ideal root structure or
like the ideotype situation of it.
Those can all be fit into this thing. I mean, we have one on campus, expert, right? He was the Wolf Prize was given at Stony Brook, that university, on morphometrics, right? Evolution actually function on the phenotype, not it's through genotype, right? You can't eat the it's a gene. You can use gene to run, right? But it's a phenotype that change that is driving the evolution, right?
Yeah.
So I thought that was relevant, so we invited him.
Well, and it makes me wonder too, like it goes back to when our conversation with class where we were really hammering out the ideotype paper in the Maze project or looking at Maze and all of its ideotypes.
Yeah.
Going back into if we were to reanalyze that using this type of a multivariate approach where we're combining all of the ideotype individual traits together in the analysis, would we start to see some that bring out more importance than others or where they're all equal because of the way we've been breeding?
Yeah. I think that's a great idea. Yeah, that's coming to my mind when I was listening to his talk. I actually listened to his video from the Roth Prize ceremony.
Oh, okay.
This is my third time listening to his talk, that comes to my mind. But I mean, then when he actually gave us when I was asking the questions about haplotype and genes, he said, well, you needed some pre-processing, right? I don't know if, if any idiotype measurement can be pre-processed. Maybe they're all already multivariate normal already, right? And then you just plug in. It needs some extra dedicated work, but only just doing research. He's actually providing the tools to make it. That's the amazing side of him,
Yeah, well, and I know Matt always, go ahead.
I'm just thinking that we may not, immediately use it right away, but it's just good to know there's something there, right?
Yeah. Well, and I think especially as we move into the genomic selection, this type of multivariate analysis is going to be critical because the whole genome isn't controlling single traits. The whole genome is controlling thousands and thousands of traits. So how are we going to investigate both of them together?
Yeah. Good.
And I need to look into his a little bit more because some of the projects that I'm working on have, like he talked about, there's a spatial and a time series component, but then also looking at like the pedigree component as well with some of our breeding trials. And this seems like it might be I need to investigate it more, but it looks like it may be a tool that'd be maybe usable for exploring some of those relationships.
Yeah, but you see that what happened that he, I'm not, as good as him in terms of the mass stats in the software tool development, but he's applying this phylogenetic relationship feeding as a mixed model to control the analysis is actually very similar or parallel to when we did this mixed model to, establish the GWAS. We feed the pedigree relationship matrix or genomic relationship to control that. What he said is that our sample are correlated, right? We don't have independent samples that are not related, right? Any correlated structure, right, of the sample has to be accounted for before you infer any distance or relationship between the genotype and phenotype. There's this this, you know, parallel between what he's doing and what we did. And then, so I thought that was so nice. Yeah. Good. Well, I'm glad that, you know, we have a few people stay until now to talk with him.
And I found this really interesting, even though it's going to take me a while to process it.
Yeah, but I was going to ask, I was challenging student. I mean, I feel that I was in this junior senior committee of Iowa State, you know, agronomy. We don't even require students take calculus 1, 2, 3, anything. I think I always encourage people to do not just stop taking courses. I actually finished the calculus 1 online myself at Khan Academy. I'm halfway of calculus 2. I took some calculus when I was in college 25 years ago, but in the last year in COVID, I just started the calculus 1. I finished it, you know, I did okay.
What I always think is interesting is that, you know, as you get farther in like your stats and your probability models.
Oh, look at that. That's
you
good.
know, we think of like stats as being like, you know, it's just a linear model, but it all eventually gets described by some form of calculus.
Yeah. So my trick is that at least if I do that, I'll pretend I'm less intimidated by when people are showing those things, you know? Okay, let you guys be busy. Thank you. Take care.
Thank you.
Bye-bye.
Bye.
Bye. Thanks, Dr. Yer.
Okay.
Bye-bye.
Bye-bye.